Hello, and welcome to our first MedTalks. I'm Steve Secker, Director of Product Management and Innovation at MedImpact, and thank you for joining us. In this new speaker series, we intend to engage with thought leaders, innovators, and disruptors in conversations on issues impacting healthcare. Quick few housekeeping items. Throughout the presentation, you will be muted, but we do encourage questions for our speaker, so please use the chat on the right of your screen. We'll try to get to as many of them as we can. And you can also use that chat if you have any technical issues. And please be sure to check out our website, medimpact.com/medtalks, for information about our upcoming MedTalk speakers. Healthcare today is changing faster than at any time in human history. We launched MedTalks to help our partners anticipate that change and harness it to improve the quality, access, and affordability of healthcare today. If you've worked with MedImpact, you know that innovation is in our DNA, and we believe it is our responsibility to promote this dialogue in the industry. Speaking of DNA, I am now truly delighted to introduce our distinguished speaker. Dr. Jennifer Doudna is one of the most brilliant scientific minds of our time, and dare I say it, of all time. To think that in our lifetime, we went from knowing the structure of DNA to sequencing the human genome, an amazing achievement in and of itself. And with that knowledge, we have begun to personalize medicine, to tailor therapies to the unique genetic makeup of a patient. But now, also in our lifetime, now with Dr. Doudna's contribution, we have entered a new era, one in which we can actually edit genes, literally rewrite the code of life. The implications for healthcare, but for many other areas, are truly profound. Dr. Doudna is professor in the departments of chemistry and of molecular and cell biology at UC Berkeley. For her work on CRISPR, she, along with Emmanuelle Charpentier, earned the 2020 Nobel Prize in Chemistry. Time magazine has recognized her as one of the 100 most influential people, and she is the subject of a best-selling book, The Codebreaker by Walter Isaacson. Dr. Doudna will be joined by our very own Mary Belanti, Managing Principal of Clinical Account Management. Mary is herself a clinician working on the front lines with our clients, developing clinical and business strategies to improve health outcomes and reduce costs. Without further ado, Mary.
Thank you, Steve. And thank you so much, Dr. Doudna. It is truly an honor to be speaking with you today.
Well, thank you, Mary. And boy, thank you, Steve, for that generous introduction. It's a real pleasure to be here today.
So in our audience today, Dr. Doudna, we have senior leaders in healthcare from health plans, hospital, health systems, and other industry players. And they are currently providing coverage for gene therapies that have come to market. Can you take a moment and explain to us, probably the question you've heard a million times, briefly, what is CRISPR and why is it so transformational for gene therapy?
Right. Well, those are great questions, Mary. And, I think we can start by saying that CRISPR is a tool for altering DNA in a precise fashion in cells. And this gives scientists and clinicians now an extraordinary opportunity not only to discover the function of genes, but also to create actionable plans for changing the underlying genetic basis for disease. So it truly is a transformational technology that is already impacting patients, and we anticipate that accelerating, tremendously in the coming years.
Can you take us back to perhaps if there was an aha moment or a moment where you realized kind of the importance of what you had discovered? Can you tell us about that?
Well, CRISPR is a great example of the potential and the power of, I would say, small science and curiosity-driven research, because the whole field of CRISPR biology began with a few microbiologists wondering how bacteria fight viral infection. And CRISPR in nature is, in fact, an adaptive immune system in bacteria. It's a way that bacteria can acquire in real time immunity to viruses, something that, boy, we could use that right about now, right? And so through studying the process of how that actually works at the level of the molecules that are involved, that was where Emmanuel Charpentier and I teamed up to determine how this immune system operates. And by doing that foundational kind of fundamental research, we realized that a protein that's called CRISPR-Cas9 is in fact a molecular cleaver. And importantly, so it cuts DNA, but here's the important part, it can be programmed trivially by using molecules of RNA that have a script that directs Cas9 to a particular DNA sequence. And it's trivial to reprogram Cas9 by changing that script. And so that really was the aha moment, was recognizing that once we had the knowledge about how this immune system protein functions, we realized it could be harnessed as a tool for altering DNA by triggering changes to genomes in any type of cell.
So you mentioned the current viral pandemic. Can you talk a little bit about CRISPR and what role it may have had in testing or some of the vaccine development?
It's interesting that in nature, CRISPR is in fact a surveillance system. So it really is a way that bacteria are naturally surveilling the genetic material that's in the cell and asking, is there something foreign here? And so we, and of course, many other teams have recognized that CRISPR could be deployed as a diagnostic tool based on its natural function. And that's where I think we're seeing the most immediate impact of CRISPR in the current pandemic is efforts underway. We have a 5-lab academic consortium in the Bay Area working on point of care diagnostics with CRISPR. And then of course there are companies and other academic groups working in this area as well. And I think that I'm hopeful that in the coming months, we will have several useful, deployed tests that are based on CRISPR that will be useful not only in the current pandemic, but importantly can be, again, easily reprogrammed to recognize either new viral variants as they crop up or other viruses when they emerge.
So if and when a next pandemic hits, CRISPR will be part of that response. That's fantastic. When did you or do you anticipate the first CRISPR-based therapies coming to market? I know we're talking about less than 10 years, but I believe there's already studies taking place for sickle cell anemia. I mean, that's a pretty quick timeline.
Yeah, it's quite extraordinary, isn't it? I still can hardly believe it. I mean, the work that Emmanuel Charpentier and I published was that published in 2012. And we're less than 10 years out, and we're already seeing the results of clinical trials with the technology, which is really, really amazing and really exciting. And as you mentioned, so one of the early targets for CRISPR clinically has been blood disorders like sickle cell disease. And part of the reason for that is, first of all, we understand a lot about the genetics of those disorders. There's obviously, very profound clinical need for patients that are affected by these diseases. And importantly, they're diseases that can be treated by what we call ex vivo therapy, where the cells that need to be edited are taken out. In the case of blood disorders, stem cells from the bone marrow, which are then edited in the laboratory and replaced into the patient. And so that just avoids the whole issue of in situ editing delivery, which is still one of the real challenges in the field. So I think we'll continue to see rapid advances, I think, in the use of CRISPR with sickle cell disease and thalassemias, et cetera. I also think on the probably near-term horizon also are eye disorders. Again, a similar reason. There are very good genetics known for certain diseases of the eye where delivery of gene editing molecules is possible, given that it's a localized kind of regional type of delivery that would be necessary. And then, maybe a little bit farther out, but still, I think, possibly in the next 5 to 10 years, we'll see real viable therapies for muscular dystrophy. I think this is also been really interesting to see the rapid advances there, where it's now possible to effectively cure muscular dystrophy in a dog-based model. And that gives many people hope that, in the next few years, it will be possible to do delivery into people that will be effective for that disease as well.
It's kind of awe-inspiring, isn't it? It's quite amazing. Question about the somatic versus germline CRISPR technology. Right now, my understanding is that it's somatic. It is in that patient only. Will it come to a point where CRISPR is used for germline, sperm cell, egg cell changes, and what kind of ethical concerns? I know you've raised some yourself. Do you have for that?
Right. Well, I think that's an important distinction to make. So right now, the vast majority of applications of CRISPR are in somatic cells. So they're in cells that are going to impact an individual, but don't create heritable changes in the genome. That's very different if we start talking about editing embryos or sperm or egg cells, where the changes then, if they were used to create for cells or embryos that were used to create a pregnancy, would then introduce changes that could be passed on to future generations, which I think is raises a lot of profound questions about, should we do this kind of genome editing? And if we do, when and where should it be deployed? Who should, decide about it? Who should pay for it? Yeah. Those kinds of questions. And I personally think that it will happen. I do, because I think that, it's a technology that, it does offer an intriguing opportunity to alter our own genes at the beginning of life. And I think that there's just an intrinsic interest in this, and so that we can't deny. Now, that being said, is it a good idea to do it? Certainly not right now, for many reasons. Part of them are technical, but there also are, I think, very important societal issues that need to be addressed before proceeding down that path. But nonetheless, it's important that the scientific and clinical and pharmaceutical, communities be already thinking about this potential, because it's not going away. I think that as I was growing up, at a time when in vitro fertilization was, kind of just coming online. I still remember, my parents talking about, gee, is it right for babies to be made in a test tube? It sounded very strange. And, and then of course we had family friends who were only able to have children through that process. Their kids were great. And my parents got used to the idea, as did many others. And, now that's a technology that's widely accepted. And I think we could see a similar kind of thing in the coming decades happening with something like gene editing as well.
So in my very basic understanding, let's just say that to start, the CRISPR technology in and of itself is relatively inexpensive or straightforward. If I'm oversimplifying, please feel free to correct me. And I know that our current experience for our audience is that gene therapies are carrying extremely large price tags in the millions of dollars per course of treatment. Do you think that the relative inexpense of the CRISPR technology will translate to lower cost of care eventually?
It's a hard question, because I feel that, as you implied, I think one of the reasons that many scientists and clinicians are excited about the CRISPR technology is that, as you said, it's widely available. It's something that, it's a tool that labs globally were able to quickly adapt for their project of interest. And that's one of the reasons that it has become such a transformative technology. That being said, as I think people here know very well, that's actually not the bottleneck for therapies, genetic therapies. It's all the other things that come into play. And let's use sickle cell as an example. This is a situation where, we have the ability to make the genetic changes that would be curative for sickle cell disease were performed back in very, very shortly after the publication of Emmanuel Charpentier and I showing how the technology worked. And so that could be done in a laboratory several years ago. But the question then is, how do you actually get it to a point where it's clinically useful and actionable? And to do that, of course, involves things like, a bone marrow transplant must be done right now to deliver edited cells into patients. we have to understand how to control hematopoietic stem cells to the, in the laboratory so that they can be edited while they're still in a stem-like state. And, a number of other factors. And so, right now, the therapy, when it's applied for sickle cell disease, costs north of $1 million, a person. And so that's clearly not going to be affordable for many people around the globe that could benefit from it. And so I think, taking a lens, my lens, of course, is always thinking about it from the science and the technical perspective. I think there's some really important, exciting advances that are happening now and in the near future that will address an access issue. One of them is better delivery vehicles so that bone marrow transplantation eventually is not required. That would be really enabling if that happens.
I know we currently have the genetic information protection in place in the United States so that people aren't discriminated against for employment or insurance. If we're talking about an era where we will be editing the genome and fixing those, does that remain relevant? Or do you still think that's necessary? And is that part of the responsible use of CRISPR?
It's a really interesting question. I mean, let's talk about a sort of hypothetical that could be a reality someday, and that is that there are risk factors for cardiovascular disease that are single genes, PCSK9, for example. That there are natural human variants of this human variation. People in the human population that have natural variations of that gene that protect them from cardiovascular disease, from high cholesterol. And so imagine that we could use something like CRISPR to give everybody that protective genetic allele. Would that be a good idea? What would be the cost? What would be the cost benefit of doing that? And, right now that's technically not possible, but I think in the future it could be. And so then that becomes a real question. Is that something that would be desirable, first of all, and secondly, how do we pay for it?
Yeah. So a specific question for you, it's kind of a personal question, and it would be, what is your hopes, your dreams for what CRISPR brings to humanity?
Oh, wow, that's a big one.
It's deep.
Yeah. I mean, I guess there are a couple of things, and you'll be surprised at my answer. I guess, and I'll say two things. One is that, as we've been discussing, of course, like many scientists, it's really humbling to know that work that I have been involved in is impacting people's lives. I mean, when I first read about Victoria Gray, who's the first patient in the US to receive CRISPR therapy for her sickle cell disease, and just how it's impacted her life in a positive way. Wow, it really touched me. And I receive emails regularly from other people who are either, thinking about how CRISPR could impact them in the future, or are looking to get into clinical trials and things like that. So, there's a lot of need out there. And so I certainly feel excited about the value that we see with CRISPR for really curing genetic diseases of that type. And then I guess the other, there's lots of other ways I could answer the question, but I guess I would like to point out that, and it goes back to what was said in the introduction, that I think that it's also a really interesting time in biology where we have genome editing technologies converging with inexpensive genome sequencing now that's increasingly available, and artificial intelligence that's being used to get better diagnostics. Sorry, I see my cat behind me. Sorry about that.
We have a visitor.
Makes it real, right? And and I think that it's really inspirational for future generations of scientists, and kids right now who are thinking about a career in science, and what area of science is going to be most exciting. I really do hope this inspires them to get involved in this really exciting kind of transformational moment in biology that we're living through right now, that I think gives so many opportunities to have a real impact on patients.
Yeah. So I know we were talking about students, and if you were looking at what's happening in the sciences right now, I know that we've talked about a wave of women and girls entering STEM professions. Is that something that you've experienced in your career? Do you see that wave happening?
Well, I've seen the wave for a long time, actually. Going all the way back to, when I was in graduate school, I was quite impressed that in my graduate class, there probably were about half women who were in my program, which was, I thought, wow, there's no gender issue. It's great. The problem is, and this has been commented on quite a bit, right, is that there's a kind of a leaky pipeline, right? And that, it sort of typically as people advance in their careers, more women tend to drop out of the professional tracks, either in academic science or in companies, for a lot of different reasons. And so I think that what I'm hoping is that, and it's change is slow. It's slower than any of us would like, but I do think that there is increasing attention paid to what those factors really are, and it's a combination of helping with family considerations, whether it's childcare or parents' care that needs to be handled at a time when for many women they become the primary caregivers, in many cases at a time when they're also under the most pressure in their professional life. And so how to mitigate that, how to create a culture that's more accepting in general of family life being kind of integrated into our professional lives. And I actually think that, there may be if there is, a silver lining or two with the whole COVID pandemic, it will be interesting to see if this is one of them, where it sort of becomes more acceptable professionally to do, talks like this, right? Where we're doing it in a virtual format, where, we have more ability to integrate our kids or our family life into our work life seamlessly, rather than requiring people to travel for things. I don't know. It's an interesting question, but I'm hopeful that actually may help accelerate the ability of women to advance in their careers now that this type of interaction is more acceptable.
Right. And you call out a very good point. I mean, the whole reason for this particular event is that our normal on-site event is not occurring.
Right.
So even for our audience members, that this is a new way, and it kind of fits into the actual workday. Speaking of our audience, of these healthcare payers and decision makers, is there anything that you would ask of this audience in order, what can we be doing to support the efforts of you and others in the field to bring these CRISPR therapies to market for patient use?
Great question, Mary. I think that, something that would be really interesting, certainly to me, and I think to a number of others who are working on the, the more upstream side of the R&D, I guess you could say, is to better understand how these technologies will ultimately be paid for. And, and I think this is something that, many people are grappling with right now. How do we think about therapy that could, we hope, be a one and done cure for a genetic disease where someone would otherwise have to take, a chronic therapy over the course of many years or even decades? How do we think about paying for something like that? And, is there a model, and I've heard different models discussed, is there a, do you have a monthly subscription, or, do you have insurance companies that will cover a cost, that's maybe a very high upfront payment, but knowing that that person is then effectively cured of their disease, and they're not going to require ongoing treatments that maybe are very, very costly over the course of many years. So I think that would be something really interesting. And the question is how to, I don't know, I always find that, it's very challenging to, break down kind of traditional silos, right? Because people like me don't necessarily often have opportunities to interact with this group here. So I think more ways of, thinking about creative ways to bring people together. And again, with virtual formats, it becomes a bit easier. And really educating, people like me about your world, and then maybe vice versa, we get more of a conversation going. I think this would be very helpful.
I agree. I'm sure our audience agrees too. And just a reminder, we will be moving to audience questions soon, so please make sure you're putting them in the chat function so that I have your most up-to-date questions. I have a kind of off-topic. What other scientific developments are you interested in right now outside of CRISPR, or is CRISPR really the only focus for you right now?
Well, CRISPR is yeah, it's kind of taken over my world, I have to say, in many ways, scientifically for sure. But the thing about it is that there are so many facets to it that are really interesting, and they intersect in ways that I couldn't have predicted. So, for example, a few years ago, we started the Innovative Genomics Institute at UC Berkeley and UCSF in the Bay Area to use genome editing to deal with real world problems, both in healthcare, but also in the environment. And so we have a growing program right now on addressing climate change, for example, and thinking about ways that genome editing is going to be impactful there. And, that's connected me to a lot of people that think about, the ethical issues around genome editing, not only in healthcare, but also in, environmental settings, thinking about how we fund this type of work. And importantly, what are the right, or really the effective ways to translate fundamental discoveries into, real world applications? It's a big challenge, and we're hoping that this institute can help to do that by, again, bringing people together that, maybe wouldn't normally talk to each other due to traditional silos, and then also finding appropriate commercial partners that can help us advance technologies when they move to a point where, it's no longer something that an academic group can kind of tackle. You want to get it into a setting where there's appropriate funding and a team that can really, run with it. And so, I do a lot of work right now to try to figure that out. And then, of course, I have my little pet academic projects that are going on. So we continue to discover new CRISPR systems. We think there's still a lot of value there, and finding different types of genome editing enzymes. We found in our work so far that all these different enzymes out of bacteria have different chemical properties that mean they're useful in different ways. For example, the diagnostics that we talked about earlier, that came from a subset of CRISPR proteins, not the original ones that I worked on with Emmanuel Charpentier. So I think there's still a lot of value there, and that's something that we focus on in our academic work.
Fascinating. So if you were to say, I'm thinking about our audience, we have 300 people approximately. If you were to have a guess, how many of us do you think will be impacted by a CRISPR therapy or a CRISPR-based treatment in our lifetime? Do you have any guesstimate?
Well, if you're asking how many of us will, have, say, a CRISPR therapy that affects like us personally or our families, it's probably going to be a small number, I suspect. And for the reasons that we discussed, right? I mean, it's still very much an emerging technology in the sense that, delivery is a challenge. Even just the regulatory climate around CRISPR is still a challenge. I mean, it's great that it's a programmable, personalizable system on the one hand, but if you're the FDA, how do you decide, does that mean that each personalized version of CRISPR for each of us has to be run through an individual trial? Obviously, that's not possible. So, I think there's still lots of debate about that. So I think for, if you're asking about, direct clinical impact or application, I think the numbers will be small. However, I think we will all of us will be personally impacted by this technology. And the reason is that I think that in the not distant future, you're going to go to your local Whole Foods or Safeway, and you're going to be purchasing products that were generated using CRISPR. And so that, that's coming for sure. And I also think that, we're increasingly going to be using CRISPR. When I say we, it's the royal we, but I think CRISPR will be used to find better drug targets, new drug targets. The pharma companies are getting involved in this now in a big way with various kinds of partnerships. And I think that will increasingly be, a very important source of, pharmaceutical discovery will be, really finding effective targets. That's still a real bottleneck in the field. So
you spoke just a moment ago about regulatory environment. Can you talk a little bit about, is it different when you're talking about, say, European Union versus US? Are you involved in the differences in the not only regulation but research in those areas?
Yeah, absolutely. It's very different. Well, it depends. I mean, I guess it depends on the area. You know, if we're talking about human embryos, yeah, it's very different. Different countries regulate that type of research quite differently. Here in the US, for example, it's illegal to use public funding for any type of human embryo research right now. But that's not true in other countries. And then if we talk about agricultural applications, you know, in the US Department of Agriculture has ruled that for many types of CRISPR manipulations of plant genomes, that's unregulated right now. They're not considered genetically modified, and they're simply not regulated. Whereas anything that has ever touched CRISPR, you know, that's agricultural in Europe is considered immediately a GMO, and it's highly regulated. So, you know, the exact same product would face quite different regulatory environments depending on where it's being marketed.
Yeah, complicates things.
It's complicated, yeah.
A question about, do you think CRISPR therapies, is there a risk that it could exacerbate the inequalities in healthcare, the social inequalities?
Absolutely. I worry about, this is the thing, you know, when people ask me, what do I worry about most with CRISPR? That's probably it, because I think, you know, like any advanced technology, it's expensive right now. It's really available only to a small sliver of the population. And unless we, I think, address that head on and think actively about how to change it, you know, it's going to remain that way. And I certainly don't, you know, want to think that work that I've done is only affecting, you know, or helping, you know, the ultra rich, right? That's not what I want. I really want it to be something that helps everybody that can benefit from it ultimately. And I know that's a long-term goal for sure. But, you know, I do think that we have to address it. We have to think about it. And you know, again, going back to sickle cell disease, I think that, you know, for us, you know, we have recently gotten an IND from the FDA. That's a 3-campus partnership between Berkeley, UCSF, and UCLA to do an initial trial for sickle cell. And, you know, why would we do this? There are already companies, you know, developing the technology for this application. Our purpose is to really look at ways to reduce cost and find ways to, you know, as I mentioned earlier, deliver CRISPR in a more efficient fashion, find ways to create the molecules that are used for the therapy in simpler, more affordable ways, and also look at ways to scale it so that, you know, just by increasing the scale, right, we could also imagine that costs will be reduced. But we're really, you know, that's really our focus with this project in our academic setting, is how do we look at the technology with a lens of affordability.
Yeah. We have a few questions from the audience. Sophia has asked a question. It's pretty profound. Certain diseases seem to actually have developed with a benefit. For example, I've read that people with sickle cell anemia have resistance to malaria, or people with Tay-Sachs were resistant to tuberculosis.
Right.
Any concern that editing the gene will introduce inadvertent side effects down the pathways?
Yeah, it's a really good question. And I think that, you know, the answer is yes, certainly, that there is concern about that, you know, and as you pointed out with sickle cell disease, that, you know, for people that inherit one copy of the sickle gene, they are protected against malaria, which is probably an important reason why that gene has been maintained at some level in the human population. Unfortunately, if somebody inherits 2 copies, then they have sickle cell disease. So, you know, they may be protected against malaria, but, you know, there's a profound sort of negative outcome there. And so what I think is important to, you know, for any therapy, really, not just CRISPR, right, is you always have to look at the cost benefit, and you have to ask, you know, is the benefit of the therapy, you know, outweighing whatever the risks might be, right? And you know, these days, you know, at least in many places, we can mitigate against malaria So it probably would be, at least for many people, beneficial to, to deal with sickle cell disease genetically if that becomes safe and sort of widely available at some point. And then, with other, taste, you mentioned Tay-Sachs. I mean, yes, I think there's a number of really interesting, biologically interesting examples of this, where a gene has been maintained in the human population because it does provide some kind of benefit, at least at a population level. And so, I think it'll always be a question of, cost benefit. Is it better to deal with that disease at the gene level or not, depending on the outcome to the patient.
We have another question from Scott, wondering, is there a potential use with this technology to tackle the long sought after goal of slowing down the aging process, advancing life expectancy? Yeah, well, Scott, you're probably not surprised to know that you're not the first person to ask me that question. And I think the way I think about it right now is that I think CRISPR, at least in the near term, is going to be very useful for discovering the molecular basis for aging. Still, that's something that it's still a big mystery. We really don't understand why we age. And, and there, it's not that people aren't interested in it, right? It's just that it's a very complicated problem and question. And I think CRISPR will be an important tool in the toolbox to start nailing down the genetics of aging, what really is happening in the aging process at the, that possibly could be altered if we altered our genetics. Mm-hmm. And so I think we're going to see, maybe, you can sort of roughly break it into two phases. CRISPR will initially be a discovery tool and try to answer those kind of really fundamental questions, but it also is a tool that can be used in a practical sense. So suppose that it were possible to, manipulate a few genes and extend what I would call health span. I don't know if it would extend lifespan, but what if we could make sure that everybody lived a healthy life? I mean, that would be huge, right? So I think, that's the way that I think about it as a therapy in the future, is more of a sort of a protective kind of therapy that might increase our quality of life as we get older. Fantastic. Well, thank you, Dr. Doudna. I know that we are coming to the end of our time. I want to thank you for being our inaugural speaker for our first MedTalks. Your insights were interesting and fascinating, and I know that your work will continue to have, well, not just a ripple effect. Really, this is going to be a tidal wave effect on our daily lives and on the healthcare industry specifically. So thank you so much for your work. Thank you for hosting me, Mary. Great to be here. And we'd like to thank our clients and our partners who have joined us today. Please take a moment and fill out our survey about today's talk. It is in your chat on the right side of your screen. And then please also be sure to check out medimpact.com/medtalks for more information and to sign up for our next MedTalks, which will be with David Oshinsky, Pulitzer Prize winning author and drug historian. And it will be moderated by our own Chief Growth Officer, Ray Marcella. But until then, everyone, please stay safe.